Yes, in the previous class you studied the one gene inheritance, then incomplete dominance, test cross, thereafter two gene inheritance, and then law of independent assortment, as well as the law of segregation we studied in the previous class. Now I will continue chromosomal theory of inheritance. You have already come across in the previous online class chromosomal theory of inheritance. Actually, this is a post-Mendelian work. In 1865, Mendel published his laws of inheritance, that is we know. And till 1900, it was not recognized by the scientist and as a genetist, because the reasons are so many. The reasons like publicity problem, because there is no communication problem is there. Like today's phone or internet or nothing was there at that time in the beginning of the 20th century or in the 1900. That's the one. Second thing is Mendel, what the blending character here was stable. According to the Mendel law, purity of gamete, and they do not blend with the one another and show stable character, that was not accepted by all the ages of the scientist. And thirdly, here the what Mendel proposed many these laws of inheritance and expression, that is by mathematical or statistical expression, that was not properly understood to the that age biologist. So because of these all reasons, Mendel work was not properly recognized. But after 1900, particularly after the 1900, in the beginning of the 20th century, the scientists like De Vries, Hugo De Vries, I think you heard, Karl Correns and Tesselmark. These are the 20th century scientists reattempted the Mendelian law of inheritance, and they also repeated the same experiment and come to the same conclusion that what Mendel did, hybridization experiment on some other plant, is correct. Of course, it not applied to all the type of plant, but instead some 5 to 10% plant have little variation in their dominance property, segregation property, and as well as in some independent segregation property. During same time, another scientist, two scientists, Walter Sutton and Theodore Boveri, in 1902, Walter Sutton and Theodore Boveri, that is called Sutton and Boveri theory, and they come to know that more and more information, because at that time cell was studied, the nuclear material was discovered, and microscope become more and more advanced. At that time the people begin to know more and more about the genetics and the cell biology. Since then people, means that age scientists studied more and more about this, and particularly Sutton and Boveri, and they also studied on the nucleus, and they come to know that the nucleus that containing the chromosome. Chromosome meaning, actually the discovery of the chromosome is by the Fleming, that is we know. But the chromosome study and the movement of the study was studied by Sutton and Boveri. And according to the Sutton and Boveri, the movement of chromosome is parallel to the movement of the gene present in the same. And they also argued that here during the anaphase 1, see anaphase 1, anaphase 1, here the paired chromosome moving to the opposite pole. That is, anaphase 1 is halvening number reduction stage it is. If it is a 46 number is there, and half number of the chromosome moving to the one pole, another pole to the another 23 chromosome. So means chromosomal number become half without a division of the chromosome. And in this stage, here what the chromosome move from the polar region, sorry, not a middle region, equator region to the polar region. At that time, along with the chromosome, some character also segregate. And in this segregation, genes also separate, means character also separate. And this separation of character during the anaphase 1 and Mendel, the gene or factor that segregate from one generation to the another generation is combinedly called as chromosomal theory of inheritance. Means in other words, exactly what we are saying is here, the inheritance material present in the chromosome. See, you know that this is a chromosome, this is a chromosome, this is a chromosome structure, and this chromosome contain inheritance material. This chromosome contain inheritance material in the form of gene. So gene is a inheritance material, and the chemistry of that gene is what? DNA. That you already studied in the molecular biology also. Molecular biology topic is also completed. So means gene chemistry is a DNA, and that DNA play a very important role here in the process of transmission of character from one region to another region, and that is what is called as the Sutton and Boveri chromosomal theory of inheritance. In other words, here all the characters, so means that is depicted in the gene, and how the chromosome move. Along with the chromosome, the gene also move during anaphase 1, and particular character that distributed to the particular cell. And that particularly you must know that anaphase 1 is a property of the meiosis, and that meiosis taken place only during the gametogenesis, not in our body cell, that to be in the testis and the ovary. So means it is only formation of gamete. So means what kind of gamete containing what type of chromosome, and in which what type of inheritance material present. On that base, the character of the individual, and that was the fundamental idea was given by Sutton and Boveri in the 1902, and that is what you call as chromosomal theory of inheritance. And this chromosomal theory of inheritance further, it is also continued by another eminent scientist, and who is the T.H. Morgan, Thomas H. Morgan, and that is called as linkage and the recombination. Linkage and the recombination. Linkage and the recombination. Yes. After Sutton and Boveri, here T.H. Morgan continued the chromosomal theory of inheritance, and how chromosomes carry the genetic character or inheritance character from one generation to another generation was correctly understood. Is it true or not? And that was further studied by the T.H. Morgan, and almost all the scientists of the 1900 and 1902, the discovery taken place, they accepted the Mendel's law. Almost all they accepted the Mendel's law, and they also come to conclusion that Mendel's law is correct, but in very few plant and animal that may be vary, and that is a different matter. But 85% of the result came again same what Mendel conducted. But after the Sutton and Boveri theory, T.H. Morgan attempted the same again, chromosomal theory of inheritance, and wherein he could identify some different characteristic feature, but his experimental the species was Drosophila. Experimental species of the Drosophila, and that is Drosophila melanogaster. This is Drosophila melanogaster, and this is commonly called as fruit fly. Generally, you can see this fly in rottening fruit. Fruit is and where you can see, and that is why commonly they are called the fruit fly. The scientific name is Drosophila melanogaster, and he used Drosophila melanogaster to study the further experiment of chromosomal theory of inheritance, and later he come to know that what the Sutton and Boveri said, the chromosomal theory of inheritance is 100% guarantee, but they come to know that here the chromosome present in what a living being is two types. There is one is autosome and another one is allosome. Here autosome means it carry only body character. It carry only body character, and allosome means it is a sex chromosome. Autosomes are generally called as somatic chromosome. Another way. Autosomes are also called as somatic chromosome, or they carry body character only. But allosomes are sex chromosome. They decide the sex of an individual. They decide what? The sex of an individual, and that is why they are called as allosomes. Autosomes and the allosome. Later, during his experiment, he come to know that when he was carrying some experiment on Drosophila melanogaster, he come to know that some character related to the melanogaster, not on the autosome, instead it is on the what? Allosome. And what he called as, it is sex-linked inheritance. Called as what? Sex linkage. Sex-linked inheritance means sometime sex chromosome also carry what? The some character from the father or mother to their sons and their daughter, means it is here the autosome not only responsible for carrying the body character, sometimes allosome also carry what? The body character, means on allosome, there are two allosomes are XX and XY is there, no? Sometimes on the X chromosome also some what genes may be located, so means that is a sex linkage, sex linked. And in the Drosophila melanogaster, what he observed, when the Drosophila melanogaster, see the yellow body and white-eyed female, yellow body, white-eyed female is crossed with brown body, red-eyed male, brown bodied, red-eyed male. And what he got, the resulting F1 generation, and what he got F1 generation, and this F1 generation when it is crossed with one another, and he did not get what? Particularly, here see yellow bodied and white-eyed female, brown bodied and the red-eyed male. So means two characters are there, yellow body capital Y, and W. Just I am giving the allele. Similarly here small y, and W. If this is a cross, what it should be? Capital Y, small y, capital W, small w. No. So these are the two different plant you will get. Both are the same plant. If this is crossed according to the Mendelian inheritance, and the F2 generation ratio should be what? 9:3:3:1. Means totally 16 plant you got, that already we deduced in a dihybrid cross ratio. So this is the dihybrid then. So this is the dihybrid. The F1 generation are intercrossed, that means kept for inbreeding, and the F2 generation you should get 9:3:3:1, means 6 recombinant you should get. But herein this cross, what T.H. Morgan did not get this type of result, and it is a completely different result he got. Why that question? And later he come to know that, so this cross made little different result is because in the Drosophila melanogaster, some character not shared by the autosome, but some character shared by allosome. In such instance, if chromosomes or the allosome chromosome if you carry the body character, and then what the result is not like this, it will be totally different. So once again here Mendelian, but the chromosomal theory of inheritance, what is there deduced by the Sutton and Bori, and little bit variation because they did not get this kind of ratio, because this ratio he got because of the autosomal linkage. But herein what this color for yellow body and white eye, color for yellow, body color yellow and eye color white, similarly body color brown and eye color red, but that character was not on the autosome. Instead of this, it is located on allosome. Because of this reason, and they have not got this kind of ratio, and they have got totally different type of ratio. Of course, plant melanogenic 16 they got, but ratio is completely different. From this experiment, they come to know that here what the sex linkage means some character also linked to the sex chromosome, and that is what we call the linkage. And recombination means some the recombination process also take place during the gamete separation, that particularly during prophase 1, that is a pachytene stage, crossing over take place, and during crossing over here they play a very important role in the recombination process. So sex linkage and the recombination, because of this failure of transmission of character from the autosome, instead of autosome, but here instead allosome sometimes may carry the chromosomal character. So that is why it is called the sex linkage. That is why it is called sex linkage. Okay, so this is about the sex linkage and the recombination. And then next one is according to your syllabus, and the next one is sex determination. Next one is the sex determination. Yes, what is the sex determination? Yes, sex determination means it is process of identification or knowing of sex of an individual. Process of identification of the sex of an individual. How it can be determined? Because you know that again we return back to the earlier one. There are two types of chromosomes are there. That is, one is autosomes and another one is allosome. As I said, allosomes are somatic chromosome. And allosome means it is a sex chromosome. When the autosome and the allosome, it is studied by some scientists, particularly Henking, scientist named Henking, who was studying on some insect, particularly on the insect. In insect, there is two type of chromosomes he observed. Some of the chromosome, they having one kind of factor, and some other chromosome they do not having a one kind of factor. What is that? He was not properly known at that time, isn't it? Here, autosomes and allosomes are also there. All autosomes are normal, and that depends if one insect containing 8 chromosome. So 8 chromosome means 6 autosome, and that is one is XX. This is female. And another one is male, if it is say XY, and this is male, correct? Totally 8. 6 autosome means A, 7, 8. 6 autosome means A means autosome, 7, 8. So this is the nuclear karyotype of any insect. Any insect, one example insect I have given. See. Here, if melanogenic, just imagine melanogenic or grasshopper or insect or anyone, and that containing 8 chromosome means here the 6 autosomes are there and they are as usual, and in both male and female it is the same, dicto same. There is no difference, same. And here in female, what he observed, what in particular in the insect, the female 2 X chromosomes are there, and in male the only X body is present, another X body is not present, and that shows genotype. You understood? Understood. So means the female that contain two, the containing 2 double X chromosome, 2 X chromosome, and that is to be considered as a female, and which containing only 1 X chromosome and another is 0, that do not contain any factor, and that is to be considered as a male. And so that is why this concept is called as X body concept by Henry. X body concept. So during the gametogenesis he observed, during the gametogenesis he observed, for example, once again I write, see, this is a 6A plus XX, so means it produces 3A plus X. This is one type OA, and this is another same. This is another type OA. So this is in what? The female insect. In female insect here, whatever the number of egg produced, all is the same, because this is a diploid and this is haploid. During gametogenesis, what here this undergone for the meiosis, and if it is a meiosis, chromosomal number become half. So means here 8 total chromosome, means here 4 should be there. In each gamete 4 is there. 3A means 3 autosome plus 1X, and 3 autosome plus 1X. So means here 100% of the ova that containing X body. Then in male you see, in male same again 6A plus XY, and herein 2 type of gamete is possible. That is, one is 3A plus X, and another one is 3A plus 0. I'm sorry, it is not a Y, it is a 0. It is not a Y, sorry, it is a 0. So means one of the some of the gamete of the male that containing X body, and some other gamete of the male that do not show the X body is a X0 type. So the organism, during the crossing, for example, during fertilization, if these two are fertilized, see, if this ova is fertilized with or united with this one, and then resulting individual is be 6A plus XX, and organism will be female. If it is this combination, see, 6A plus X0, and that will become a male. So means here during the fertilization, if the X0 is there and it is a male, if it is a 2X chromosomes are there, that is to be considered as a female. And this is how determination of sex, later on studied by Henking and many more the scientists, and this is also studied same in human being also. In human being also, determination of sex it is studied same. In human being, you know, the total number of chromosomes present in a cell is called 46, and during gametogenesis, here in a male or female, and that gametes are formed. See, 23. I have first of all I have to write nuclear karyotype 44A plus X, and this is of female, and then 44A plus XY, and this is of male. Okay, see, these are the two different type of karyotype, male and the female. This is a female and this is a male. And see, during the gametogenesis, the female produce 2 type of gamete, 22A plus X, and here is also 22A plus X. Correct? Both are the similar type, and that is why they are called as homogametes. Homogametes. These are called homogametes. But in male, you see, during gametogenesis, 22A plus X is one type sperm, and another one is 22A plus Y sperm. So means it is a heterogamy. ಯಾಕಂದ್ರೆ ಇಲ್ಲಿ ಎರಡು ಡಿಫರೆಂಟ್ ಟೈಪ್ ಗ್ಯಾಮೆಟ್ಸ್ ಬಂದಿದೆ, ಒಂದು ಎಕ್ಸ್ ಟೈಪ್ ಬಂದಿದೆ, ಇನ್ನೊಂದು ವೈ ಟೈಪ್ ಬಂದಿದೆ. ಬಟ್ ಇನ್ ಫೀಮೇಲ್ all are the same type, because it contains double X, but in male it is a one X and one Y. So now this is during gametogenesis. In the gametogenesis, here sperms are formed and here eggs are formed. Okay. Now during fertilization, how determination of sex is judged, or how the determination of sex is taken place? If see this ova if fused with this sperm, just imagine 44A plus X again diploid is maintained, so means it is what? Female. If it is this one is combined with this sperm, and then it is 22A plus XY, so means it is male. Now I think, 44A, 44. Now I think you understood how exactly the zygote formation taken place, and that is due to union of male and the female gamete. See, this male gamete and the female gamete. This is a male gamete and the female gamete, and this is a sexual determination in the human being. In human being, so like this, the individual sex is determined, and that is why you must remember that here male play a very important role in determination of sex. If any couples they don't have a male child, the blame should not go to the mother, because mother is not responsible for giving a male child. Father only is responsible for giving male child, because the father's sperm are two types. There is one is X type, another one is Y type. So Y play major role in determination of male sex. So that is why if this Y is very strong and more viable, then it can meet with the one of the female gamete, any one of the gamete, either this gamete or this gamete, any gamete, and male child will be born, because the male gamete is only motile. And so that's motile. So means that during the discharging of the sperm into the female genital duct, if the Y chromosome reaching first to the destination of ova, and then Y fused with one of the female gamete, and the child will be the male. If no, it is not like that. If the X type of sperm, X chromosome containing sperm is more motile, more viable, and then reaching first to the ova, and then the child will be the female. So that is why you must remember that the determination of sex is not in our hand, and so that depending upon the viability, motility, and strength of the sperm, which is reaching first to the destination of the egg, and that to be the isthmus junction, that you know, and that to be fertilizing area. So in this way, your male play a very important role in the determination of the sex.